Hi everyone, welcome to the NOAA Omics Seminar Series. We'll get started in about 5 minutes. Hi everyone, and thank you for joining today. Welcome to the NOAA Omics Seminar Series. I am your host, Nicole Miller. I sit in NOAA Ocean Exploration. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the past few years. We have established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside NOAA. These seminars from these series are made available on our YouTube and posted on our NOAA Omics landing page. There should be a link to the website in the chat now. As participants, you are muted, but feel free to type questions into the questions for staff box throughout the presentation. And at the end, I will read the questions aloud for the presenter to answer. So with that, our presentation today is titled Understanding the Genetic Bias of Runtime Diversity in 4 Species of Pacific Salmon by Dr. Wes Larson. I'll go ahead and share screen presenter. Alrighty. Wes received his bachelor's in science from the University of California, Santa Cruz, and his PhD from the University of Washington. His PhD focused on applying genomic tools to study local adaptations and inform management of Pacific salmon in Alaska. Wes is currently the program manager of the genetics group at the NOAA Alaska Fisheries Science Center. Wes's research interests include all things omics with a focus on population genomics, genetic stock identification, and developing meta-barcoding tools for eDNA and mixed DNA sample applications. Thank you, Wes, for presenting today. And I'll now pass it off to you.
Perfect. Can you hear me okay?
I can hear you and I can see your slides.
All right. Well, thank you everyone for coming. I'm really excited to talk about some research we've been doing that we think is pretty neat. Trying to understand the genetic basis of runtiming diversity in a few different species of Pacific salmon. So before I get in the presentation, I just want to thank my co-authors, especially Pat, Natasha, and Dave. It's really a pleasure to collaborate with colleagues like this, and yeah, I just really appreciate all the co-authors on this research. Okay, so no surprise to anyone, but migration is a hugely important component of many organisms' life histories, whether it's large migrations of buffalo and things like that in Africa, or salmon. And so migration is just a really important component of life histories that is a large determinant of reproductive success and fitness in many species. And so getting into salmon, salmon, as everyone knows, I'm sure, travel out to the ocean and feed for usually like 1 to 6 years, and then they come back to spawn. And their timing of migration is time to give offspring the best chance of survival. And salmon display a large diversity of runtiming strategies, and this runtiming diversity creates more stable ecosystem services and increased opportunities for consumers. And so what determines runtiming? Well, runtiming is highly correlated with temperature, because temperature influences the development rate of juveniles. Therefore, salmon from warmer streams will develop faster than those from colder streams. So the graph over here is from a system of lakes in Bristol Bay, and on the x-axis is stream temperature, summer stream temperature, and on the y-axis is salmon spawning date. And you can see that there's a very high correlation between stream temperature and spawning date. And so in general, colder streams spawn earlier than warmer streams, and that's because in colder streams, the juveniles take longer to develop, so they have to spawn earlier so that they outmigrate at a similar time as the warmer streams. In other words, the theory is that they're all trying to sort of emerge at a similar time to take advantage of like resources and food availability in the spring. And so they time their spawning to make sure that their offspring come out around the right time. So in other words, salmon spawn at different times so that their offspring can emerge at a time when resources are plentiful. And there's some really different run timing strategies in salmon. In some systems, especially at lower latitudes, salmon spawn across a large portion of the year in discrete runs. Just to go back to this graph, you can see here, these are just streams. There is a little bit more diversity in spawn timing, but in general, in Bristol Bay, which is you know, in Alaska, like sort of Southwest Alaska, the first streams start in early July, mid-July, and then the last streams start in kind of late August. So you're talking like 6 weeks. There's some other environments that spawn a little bit later than that, but basically you're talking like 2 to 3 months. But in more southern latitudes, especially in places like the Sacramento River, salmon can be spawning almost all year around. Okay, so salmon spawn across large portions of the year in these discrete runs, and these different runs can take advantage of different conditions, i.e. different thermal or flow regimes. And diversity in spawn timing can also increase habitat carrying capacity as fish are using resources at different times. And the Sacramento River is kind of in particular really interesting because these different runs have kind of evolved to take advantage of these different flow regimes to be able to enter habitats that might only be passable at certain times of year and things like that. So there's just this really amazing diversity of run timing that's kind of evolved in response to the kind of Mediterranean climate that's down there and the long growing season and really unique kind of hydrodynamic regimes and things like that. And so I should say, like, that's largely for Chinook. This little graph here is for Chinook. And so most of the work that's been done on the genetic basis of run timing has been in Chinook and steelhead. Well, pretty much all of the work. And these Chinook and steelhead have these discrete runs, with earlier runs entering freshwater in a more immature state and ending and spawning at different times. And so some runs will come in and they'll be like what we call silver bright. So they look like they're really like chrome silver, and they come in and they pass on these like high flow times, and then they kind of just hang out in the river for months and months and then end up spawning quite a bit later. And quite a bit of work has been done on this over the past 10 years or so, and it turns out that there's a major effect locus called Greb1L that's highly differentiated between these very discrete runs of Chinook salmon and steelhead. And so for Chinook salmon, it's spring versus fall, and steelhead, it's called summer versus winter. And these are just graphs of allele frequency differences between spring and fall Chinook and summer and winter steelhead. And you can see that there's almost fixed differences between these different runs at this Greb1L locus. And the Greb1L genotype accounts for between 46 and 85% of variance in run timing. And different evolutionary events led to the variation in each species. So you can see that the shape of these kind of peaks is quite a bit different. In Chinook, it's quite a bit broader. In steelhead, it's a little bit kind of narrower. And that mixed with kind of doing phylogenetic trees of this specific gene makes it pretty clear that the variation at this genomic region evolved multiple times separately in Chinook salmon and steelhead. Okay, so quick review of run timing. Oh, and this is kind of a cool graphic from the Klamath. So the top here is the kind of the silver bright, and so it says with months to wait until it's time to spawn, springers enter the Klamath relatively immature, packed with energy reserves that make their flesh dark red and rich in flavor. And so they're silver, they've got really dark flesh, whereas the fall run enters a lot more more like what it's going to look like when it's about to spawn. And so for many systems and species like sockeye salmon in a system of lakes in Bristol Bay, run timing variation is continuous and related to temperature. But for some species, there's these discrete runs that enter freshwater at different times and spawn at slightly different times. And nearly all of the previous genetic work on run timing has been done on these discrete runs. And this has been a major focus of conservation genomics for the past decade. So specifically, whether or not these different runs should be kind of given specific conservation protection based on, yeah, whether like spring and fall runs in a given stream should be given sort of different conservation statuses has been a major kind of topic of discussion for the last decade or so. Okay, so what about run timing in other salmon species? And this was pretty fun. I actually was able to find pictures of my son with every other Pacific salmon here. So pink, chum, sockeye, and coho. So our goal was to investigate the genetic basis of run timing in coho, chum, pink, and sockeye salmon. And here's our study populations. And so pink salmon, there's a zoomed in version of this on the next slide, but just kind of an overview. So pink salmon, we had a population in Southeast Alaska. For coho, we had 2 populations on the Kenai Peninsula. For sockeye, we had 3 populations in Bristol Bay. And then for chum, we had a summer and a fall run chum in the Yukon. And so here's kind of a more zoomed in version of that. So the first one here is for pink salmon. We were studying a population in Ock Creek, which is near Juneau, and there's a weir on this system, and it's been studied for over 40 years with quite a lot of research on run timing in the system. And so on the bottom here, we have kind of where all of the different samples actually ended up spawning. So for pink salmon, the early part of the run spawns in kind of early August, and then the late part of the run spawns in mid-September. So we got samples from kind of early August and mid-September that we were comparing. For sockeye, we had 3 populations in the Wood River Lakes in Bristol Bay. So teal is a cool, like a colder creek, and that spawns in kind of mid to early August. Whitefish is a warmer creek, and that spawns in kind of late August. And then anvil is a beach population, so they spawn on lake beaches, and they spawn in mid-September. The next one's for coho. There's some pretty interesting run timing variation in coho in the Kenai River. And so these populations are from Skilac Lake outlet and inlet. And one of these populations spawns in kind of like early September, which is relatively typical for coho salmon. But the other population spawns in late January, which is pretty unique. And we think it's because of some kind of groundwater input that keeps it warm in that area, but we're actually not super sure exactly why they're doing this. And then the last one is chum salmon, which it's kind of easier to see from this. So chum salmon in the Yukon River, in the lower river, chum salmon spawn in the early summer, kind of early to mid-summer. And then in the upper Yukon River, chum salmon, and this is the longest migration for any population of chum salmon. They migrate thousands of kilometers up the Yukon River, and they spawn in the early fall. And so that's those are the 2 populations of chum salmon that we're comparing. And here's the kind of just an overview of the sample sizes for the whole genome sequencing that we did. So we did low coverage whole genome resequencing. We have some kind of variation in coverage, but usually, so let's see, for chum, it's around 1, coho, it's around 1, sockeye, it's more like 3, and then pink salmon, it's around 2, so 2x coverage. And so basically, we did whole genome sequencing on 30 to 50 individuals per species per run timing, and we tried to look for areas of the genome that were associated with run timing. Okay, so here's the genome scans of run timing, and we aligned everything to the chum salmon genome just to make it easier to kind of compare. And chum salmon are sort of phylogenetically intermediate, and so we thought that was a good choice. And so basically, just to make it easy to look across. And on the y-axis here is FST, so that's a measure of genetic differentiation. And on the x-axis is each chromosome in the chum salmon genome, and the different chromosomes are kind of have alternating colors here. And what we saw is there's two kind of major spots that seem to pop out repeatedly. This one here on chromosome 35 in pink, sockeye and chum, and then another one on chromosome 29 in coho and chum. Okay, so zooming in on those two chromosomes. So chromosome 29 is the one that has that peak in coho and chum, and they look pretty different. Again, kind of like the grub 1L, where in coho it's quite a bit wider, but it is centered on this what we're calling this ESRB gene, this little blue spot right here. And then in chum, it's tighter, but again centered kind of on this this blue ESRB gene. And then in chromosome 35, each species looks quite a bit different. So sockeye is kind of the most broad, like the longest sort of peak, or the widest. Chum is kind of intermediate, and then pink is pretty tight. And those ones are all centered around this LRRC9 gene, which is the pink gene right here. Okay, so what are LRRC9 and ESRB? And just to make sure that it's pretty important, so I just want to make sure that everyone gets it. So these are all the genes in these kind of these regions that are showing high genetic differentiation between run timings. And for the 29, it seems like it's centered around ESRB, but it is broader than that. And then for 35, it's kind of centered around LRRC9, but again, it's broader than that. Okay, so what are LRRC9 and ESRB? LRRC9 is a leucine rich repeat containing region. And these are common structural components of proteins. And LRRC genes are involved in many cellular processes, including regulating gene expression and immune responses, but the function in salmon is unclear. ESRB is an estrogen receptor. It codes a nuclear receptor transcription factor, which is activated by estrogen, and it's involved in diverse processes, including metabolism and heart function. And again, the function in salmon is unclear. Okay, so now that we kind of found these regions and we started to look at the genes, the next thing we wanted to do is try and genotype each individual and see if we could look at the difference in frequencies, allele frequencies between the different samples. And so these are PCAs, principal component analyses of just those small regions that are highly diverged between the run timings. And essentially what you see is for each one of them, you see that on the left, that's one homozygote. In the middle, those are heterozygotes, and on the right is the other heterozygote. And so you can call genotypes by basically using these PCAs to create cutoffs, and then just calling the genotypes based on that. And so that's what these graphs show. And you can see that for like coho, for example, there's very few heterozygotes. There's only one heterozygote here at ESRB, whereas for pinks, for example, there's quite a few heterozygotes. And the other thing we wanted to do is we wanted to check and see if these were independent evolutionary events in each species. So we made phylogenetic trees with just these regions, just the ESRB and the LRRC9 regions. And you can see that it's, we had to actually like use these little, these like dashed lines here, because when you make the full tree, the length of the branch between species is much higher than the length of the branch within species. And so basically, the divergence between species at these genes is much higher than the divergence within species, which tells us that the variation at these areas was happened independently in each species and didn't come from kind of the same variation. So again, these are independent evolutionary events in each species, just like with the grub-Linnell. Okay, and so here are the LRC9 and ESRB genotype frequencies in our populations. So we have pink salmon here, and I will say, so we sequenced pink salmon from the even run, but we also did some genotyping of pink salmon from the odd run to see if it the pattern held true. And we'll get into pink salmon a little bit more later, but essentially pink salmon have an obligate 2-year life cycle. So they're temporally segregated, and there's no gene flow between pink salmon that spawn in alternating years. And so they're a really nice kind of system to look and see, like, if kind of the genetic basis of traits is kind of similar across these different lineages. And so anyway, we looked at both pink even and pink odd, and you can see that there's kind of a similar pattern, although it's a little bit more striking in the even, with mostly early homozygotes and then more late allele homozygotes and some heterozygotes in the late populations. In sockeye, we see that the early stream is mostly early allele homozygotes. The late stream is kind of almost a third of the early allele homozygotes, late allele homozygotes, and the heterozygotes. And then the late beach is almost completely the late allele homozygotes. And then in chum for LRC9, we see that the summer run is mostly early allele homozygotes with some heterozygotes and a few late allele homozygotes. But interestingly, the fall run is fixed for the late allele, so there's no other genotypes in that one. And when we go to chum in the ESRB, you can see it's almost the same, where the late only has like a couple heterozygotes, but it's mostly just that late allele. And then finally in coho, the 2 runs are almost completely fixed for the different alleles. There's like 1 heterozygote in the late population, but otherwise it's almost completely fixed for alternate alleles. Okay, so some takeaways so far. There's 2 small genomic regions that are strongly associated with run timing in 4 Pacific salmon species: LRC9 in chum, sockeye, pink, and ESRB in chum and coho. The functional significance of the regions is unclear. There's few non-synonymous mutations that change amino acid sequences. So I didn't say this in any of the slides before, but essentially when you look and see if the mutations actually change, the mutations that we find in these regions actually change the amino acid sequence of the genes, there's very few of them. So most of the mutations are in kind of non-coding. They're either non-coding or they don't change the amino acid sequences. The early and late alleles look like they arose independently in each species. And divergence differs among species. So the chum and coho are nearly fixed for different, have nearly fixed differences between runs separated by substantial space or time. And the pink and sockeye show differentiation on very small scales. Pink separated by weeks in the same creek, and then sockeye separated by weeks and months in proximate habitats. And so just to kind of go into that a little bit more. And so the chum and coho, especially the chum, are separated by thousands of kilometers, and they spawn a couple months apart. But the pink on the other extreme, I mean, it's one small stream, and these salmon are showing massive differences in allele frequencies on the order of a couple weeks in a tiny stream, which is pretty unbelievable to think about. Okay, so it's going to get a little bit more complicated, but a little bit more interesting here. So it turns out that LRRC9 and ESRB are duplicated versions of each other. And so this is chromosome 29 and this is chromosome 35. And these different lines show what's called the homologous regions between the two chromosomes. And it turns out that the region, the ESRB region and the LRRC9 region are duplicated versions of each other. And hopefully it's going to make more sense after explaining how salmon work. So for salmon people, this will be old news, but for other folks, it's kind of crazy to think about. So salmon underwent a whole genome duplication about 80 million years ago between when they diverged from pike and when they speciated into other salmonids. And so what that means is that there's two versions of every chromosome, and some of the chromosomes are still are not fully rediploidized and are still recombining, and some of the chromosomes are fully rediploidized. These two chromosomes, 29 and 35, are fully rediploidized, but they are the same version of each other. So these regions contain the same genes, but they've been allowed to diverge over the last 80 million years or so. Okay, so let's see. Chromosome 29 and 35 are duplicated variants of the same ancestral chromosomes. For salmon nerds, they're not homeologs, they have rediploidized. And both chromosomes contain the duplicated variants of the same genes. Okay, so what about the evolutionary importance of genome duplications? There's been quite a bit of kind of theoretical work done on the evolutionary importance of genome duplications, and kind of what the conventional wisdom is that genome duplications is an opportunity for genes to change function. So, for example, if you all of a sudden have two versions of the same gene, you may only need one of those genes to actually do its job, and the other one is free to kind of evolve a different function or kind of change expression or change dosage or things like that. And so it's thought to be a pretty important evolutionary process that can create kind of new adaptation and things like that. And so multiple versions of the same gene can also be maintained and expressed differently, resulting in different dosages, which is also important. And so our best guess is that since both of the genes in these different regions are involved in run timing, it seems unlikely that they have neo-functionalized, which means that they have evolved a new function. It seems more likely that variation at both genes is maintained to regulate dosage. For example, variation at both loci is potentially required for the unique fall run phenotype in chum salmon. So in other words, both of these genes are showing very, very high differentiation in chum salmon, and that may be necessary to see this like really diverged phenotype between the summer and fall run. Although that is just kind of a hypothesis, we can't prove that, but that's kind of our best guess. Okay, so going broader, I wanted to kind of put this in the ESRB and LRRC9 in the context of kind of other major effect loci and kind of in general how kind of the relationship between genotypes and phenotypes. So most phenotypes are controlled by many genes of small effect. That's kind of the classic conventional wisdom. Some phenotypes have a simpler genetic basis controlled by large effect loci. Things like single or a few mutations that are involved in stickleback armor plates, or like mice or rabbit coat color. There's also things like large genomic regions protected from recombination, like inversions that control sunflower ecotypes and cod ecotypes and things like that. And then finally, there's things like master regulatory regions like Hox genes for body shape and tumor regulating genes. In terms of large effect loci in salmon, it seems like the kind of general rule that the large effect loci in salmon influence complex phenotypes. And so these are the kind of the previously discovered, along with ours, large effect loci in salmon. And so OMI5 is a large inversion associated with anadromine and steelhead. VGLL3 is a small genomic region associated with age and maturity in Atlantic salmon. 6.6 is similar to VGLL3. It's associated with age and maturity in Atlantic salmon and steelhead. Grebonell is the one we talked about earlier. It's associated with run timing in Chinook and steelhead, and then our LRRC9 ESRB. And just to kind of clarify, when I say small, I mean kind of under a megabase. And so besides OMI5, which is massive, it's like 40 megabases. All these other ones are under a megabase, you know, somewhere in like the 200 to 700 or 800 kb kind of range. And so characteristics of large effect loci in salmon. There's a few large effect loci that are found in multiple species. Grebonell in Chinook and steelhead, 6.6 in steelhead and Atlantic salmon, LRC9 in pink sockeye and chum, and then ESRB in coho and chum. And again, there's only one large region, OMI5, only one kind of really important inversion, and the other ones are these kind of really small 200 to 700 kb regions. And the loci in multiple species erodes through independent evolutionary events. And it seems kind of the general pattern here is that there's few non-synonymous mutations, and the region of highest association in these major effect loci or these large effect loci is often intergenic, so it's not necessarily in a gene or right on top of a gene. So how are large effect loci in salmon named? So this is in Atlantic salmon from Larson et al. 2015, and this is the region that was named 6.6. But as you can see, there's actually a much wider association than just right on 6.6. And actually the peak of association, which is this little red thing, is not even in 6.6. And notably, LRRC9 is right over here, and there is a pretty high association there between LRRC9 and this is age at maturity in Atlantic salmon. And so it's kind of the same with ours, right? We sort of named our LRRC9 one based on the fact that it seems like that was kind of the peak of highest association, but there's a much broader kind of area of association that includes many more genes. So again, the highly associated loci often span multiple genes, and importantly, 6.6 is in the same region as LRRC9 and ESRB, which is pretty interesting. So again, 6.6, which is a family, which is part of a family of transcription factors, is in the same region as our ESRB and LRRC9. And 6.6 is involved in reproduction, eye development, and other processes, and it's highly associated with age and maturity in Atlantic salmon and steelhead. And the duplicated, the duplicated 800 kb or so region that we identified contains 3 of the 6 large effect loci documented in salmon to date. So it includes 6.6, which is age and maturity in steelhead and Atlantic salmon, and then our LRRC9 and ESRB. So we think that these large effect loci in salmon are likely master regulatory regions. So again, these regions are relatively small, less than a megabase, and they're not inversions. There's few non-synonymous mutations, and they often span genes and intergenic regions, so they're not sort of focused right on a gene. They're linked to highly complex phenotypes that are controlled by many physiological pathways, and they probably control the expression of many genes through simple genetic architecture. And so there really hasn't been a lot of functional work done on any of the major effect loci in Pacific salmon. Really, the only functional work done on major effect loci in salmon is the VGLL3 gene in Atlantic salmon. And it's some pretty impressive stuff. This is out of Craig Primer's lab, and Juka has quite a few papers on this, but this one is, I think, especially interesting. So the paper is called "A Complex Mechanism Translating Variation of a Simple Genetic Architecture into Alternative Life Histories." And they did a whole bunch of like functional genomics and RNA-seq and that kind of stuff. And the results reveal a genetically simple yet functionally complex architecture where a single major effect gene produces pleiotropic variation in the spectrum of cellular traits. And pleiotropic basically means that like kind of one gene influences a whole bunch of other genes. And they kind of, they end by saying here that it's essentially a surprisingly simple genetic architecture. So this one gene, or this one kind of small region, influences a whole bunch of different cellular processes and pathways and things like that. Okay, so conclusions. 2 small genomic regions are strongly associated with run timing in 4 Pacific salmon species. And the early and late alleles arose independently in each species. And LRC9 and ESRB are found on duplicated versions of the same ancestral chromosome. A previously discovered large effect locus 66 is also found in this region. And lack of non-synonymous mutations in the presence of transcription factors suggests that this is a master regulatory region that has pleiotropic effects on many genes and biological processes. I think it's important to think about why this evolved. And so there's got to be a good reason why in salmon the evolution of like a single region controlling such a complex trait evolved. And we think it's because this allows like extremely rapid evolution and rapid changes in this phenotype based on kind of the environment. So for example, if there's a good reason to like change your run timing, for example, you should be able to change it relatively quickly within like a couple generations or even a generation, because it's only controlled by a single gene. Whereas if it was controlled by sort of a whole bunch of different genes, it would take much longer to evolve. And so we think that at least for the run timing, the reason that salmon have evolved kind of this very simple genetic architecture is so that they can kind of adapt very quickly to differences, to sort of changing environments and respond very quickly, kind of evolutionarily. Okay, so what? So our findings provide evidence that small genomic regions can influence multiple complex phenotypes across many species, challenging prevailing evolutionary theory. This is also the best example of adaptive importance, the adaptive importance of whole genome duplications, the whole genome duplication in salmon. And again, the simple genetic architecture should allow populations to respond extremely rapidly to environmental changes. Maintaining variation at large effect loci is vital to preserve the portfolio of diversity and ensure populations are able to adapt to changing conditions. And it's important to kind of take these loci into account for propagation and conservation programs to make sure that the correct run timing genotypes are kind of matched when restoring populations. Essentially, these run timing genotypes are probably a very important component of fitness. And so for conservation and restoration, ensuring that the run timing genotypes are matched to the environment is really important. Okay, so where are we going? So we've done all this work to try and find these loci and characterize them. But where we want to go next is kind of move away from whole genome sequencing and into just genotyping these loci with a few either GT-seq or TACMAN assays to kind of get a really good, on many individuals, to get a better idea of how they're functioning in wild populations. And so we plan to do single SNP genotyping of pedigreed populations, look at temporal collections to see how allele frequencies have changed across time, and look at populations across the range. We also want to test the hypothesis that frequencies change quickly and are associated with the environment. because we think that LRC9 and ESRB and other major effect loci could facilitate rapid adaptation to climate change and other selective pressures. And just to conclude here, I want to talk a little bit about some of the work that we're starting to do kind of on this research theme. So this is with pink salmon, and so pink salmon have, again, I mentioned it before, have this obligate 2-year life history with even and odd year lineages that are temporally separated but often spawning in the same system. So it's this really interesting natural common garden. So basically think about a situation where you've got a creek, and in alternating years there's pink salmon that are using the exact same environment but are totally genetically different and have been separated for tens of thousands of years. And interestingly, the even and odd lineages show very similar run timing despite very different genetic backgrounds. And so this is an example of strong determinism, which means that similar selective pressures have led to similar phenotypes. And Krista Oak has a really nice paper on this from 2019 that's a great read. So we wanted to see, are run timing similarities by stream facilitated by LRC9? Do even and odd lineages show similar LRC9 frequencies for a given run timing? And do LRC9 allele frequencies vary consistently by run timing across the range? And so big thanks to the Alaska Department of Fish and Game for genotyping these populations. We integrated 6 loci from the LRC9 region into a GTC panel that they were genotyping for baseline development. And they genotyped them on even and odd populations from Russia, Western Alaska, kind of Central Alaska, and Southeast Alaska. So 21 collections, 12 even, and 9 odd. And so on the y-axis here is just the frequency of the early allele, and then on the x-axis is the calendar day. And the calendar day is just the day that these fish were sampled. And so the relationship could even be tighter if you were able to get a good phenotype on exactly like what maturity they were, but it seemed to work pretty well anyway, even with that. And so there's a very strong relationship between the date that a pink salmon was sampled and its LRS9 frequency. So for example, in early July, those couple Russian populations and that one in Western Alaska, they're nearly fixed for the early allele. And then contrastingly, the populations in Southeast down on the bottom here are nearly fixed for the late allele in early October, late September. And then it's sort of a gradient throughout, but we were pretty surprised by how tight this relationship is. And so what this indicates to us is that, number one, LRC9 is working pretty much the same way in even and odd, despite the fact that their genetic backgrounds are totally different. And two, that there's likely not many other loci that are influencing run timing at all. So it seems like it's pretty much largely being driven by just the LRC9, although we still need to do some more work to kind of partition that variance out. But anyway, it seems like there's a really strong relationship between sampling date and run timing that holds true across lineages. And we'd like to try and look at making similar graphs like this for other species, the other species, to see if they hold true. But we're pretty excited about this result in pink salmon to start. All right, that's all I got. Thanks to all the field crews out there that collected all these samples, to ADF&G for genotyping and sharing samples, some great conversations with some really good folks there. And with that, thanks for listening, and I'll take any questions. And this is Oak Creek. It's a pretty sweet study site, like 5 minutes away from our house, which is always pretty cool. But yeah, thanks so much for listening, and happy to take questions.
Awesome presentation, Wes. Thank you so much for this very interesting work. Lots of exciting findings to come out of this work over the past few months, and very excited to see where it goes. So we'll open it up to questions. I'll get us started, and then we'll move forward. So my question is, you had mentioned at the beginning of your presentation that with runtime, there's a correlation with temperature. And then you'd also moved into with regulations in the LRRC9 and ESR right, you're seeing differences of these populations based at these two genes. And I'm wondering if you have any suggestions about the role that LRRC9 and ESR might be linked as far as temperature regulation, especially because temperature can be correlated with runtime. Does that make sense?
Yeah, I think I see where you go. So one thing we'd want to test is like whether or not there's different developmental rates associated with the different LRC9 or ESRB genotypes. But yeah, I guess I don't know about temperature regulation. I don't really even know how salmon regulate temperature.
Well, I was just wondering if there were any correlations. I mean, there are correlations with runtime, right, and water temperatures. And so looking at these genetic variations in regard to these temperatures might be something interesting to consider.
Yeah, I mean, the mix between temperature and run timing in these genes is definitely really interesting for sure.
Yeah, especially because you had mentioned too, right, that we might see increased variations with changing temperatures. Is that and changing climate temperatures? Maybe.
Yeah, I think we'd like to do some kind of maybe like a modeling exercise and try and see like you know, you might be able to project what the allele frequencies would look like into the future, and there's a point where they can't change their genotype anymore. Mm-hmm. So I think that'd be interesting to get into to see like, yeah, if like they're at the end of their kind of adaptive potential, basically.
Cool. Well, we do have a few more questions coming in, and we'll move into those. So this one's by Greg, who says, if pink salmon runtime is controlled largely by LRRC9, you'd expect that the intermediate populations would have higher variation in runtiming, since they would be segregating for LRRC9 alleles compared to more extreme populations, which are fixed. Do you find that's the case?
That's a great question. I don't think we can say for sure yes or no, but I think that could be pretty likely, based on like little glimpses of data here and there. It's actually like data on runtiming is pretty tough to get, but based on like little snippets here and there, I think that could be reasonable.
Thanks. All right, our next question is from Reed, who says, hi Wes, thanks for the great talk and cool science. 2 questions. I'm sure you've checked this, but is there evidence for inversions for any of your outlier regions? And I'll mention the second after.
Nope. Yeah, so these do not look like they're inversions.
And the second question is, for these run timings, which are relatively discrete, these large effect loci make sense. Under continuous variation for run timing, do you see or think you'll see a more polygenetic bias?
So funny enough, it may be the opposite. So pink salmon are the most continuous, but may have the simplest genetic basis to run timing of all these species. So this is kind of where we're going, but I think pink salmon, like they actually have pretty protracted runs, and I think that's kind of an evolutionary mechanism for them to like spread their spread their risk. So funny, like totally counterintuitive, but surprisingly, the continuous ones seem like they have the most variation.
Mm-hmm. Thanks. All right. So we have one final question today from Cheyenne, who says, awesome talk. You mentioned that there are a few NS mutations in LRCC9 and ESRB, and therefore you think that the signal you see is driven by regulatory or non-coding elements. Did you build a tree or look into the flanking non-coding sequences? Mostly wondering if you're sure that these regulatory regions evolved independently.
Yeah, so we built a tree. We're pretty sure the divergence between species is like orders of magnitude higher than the divergence within species among like between alleles within species, which seems to be the pattern for the other major effect loci in salmon as well. So I know it's kind of crazy to think that they would have like not used the same variation, would have evolved independently, but it seems like that's the case, which is, yeah, pretty interesting.
Mm-hmm. Oh, looks like we have one more. Thanks for that response, Wes. This one's from Monica, who asks, do you have information on the origin, hatchery or natural origin of the different samples? As I understand it, there's a large presence of hatchery origin pinks in the North Pacific. Do you have any thoughts on how this may be interacting with the results for pink, but also other species?
Yeah, that's a great question. So there's no hatchery fish in any of these samples. But we would like to explore this in Prince William Sound, where there is a bunch of hatchery fish, which I think would be really interesting. So yeah, for here, no hatchery fish, but understanding the dynamics of this and kind of how that goes with the kind of hatchery wild interactions, I think could be a really valuable line of research.
Awesome. Well, with that, Wes, thank you so much for joining us today. I also thank all the participants that were able to join today. And our next seminar series will be this December 12th at 2 p.m. Eastern Time by Gregory. And it will be on the Great Lakes Atlas of Multi-Omics Research, or GLAMOR, a new database to come out for Great Lakes Omics. Wes, thank you so much for joining us today, and I hope everybody else has a great rest of your week.
Thanks for coming, everyone.